Because cells are colorless and transparent, they're practically invisible to the naked eye, even with microscopes. And so contrasting improving techniques are used so that we can recognize the cells and their components. These four are the techniques that I'm going to be talking about: chemical stain and dyes, enzyme labels, fluorescent labels, and electron dense labels. I'm also going to quickly mention two methods for unstained cells, so we can see cells without staining them. This first example is a phase contrast, and this other example is called differential interference contrast, or Microscopy. This is the same cell, so squamous cells from a buccal smear, so from scrubbing the inside of your cheek. And as you can see, you can see them, but it's not really that clear or anything, so I'm going to talk about those techniques. Chemical stains and dyes are usually used in pathology, so for diagnosis of disease such as cancer. And in this example, we have hematoxylin and eosin, H and E. What they do is that the hematoxylin dyes the nuclei blue. So you can see these blue little dots over here. They're all nuclei of different cells, like these little blue dots. And the eosin dyes everything else in a pink, reddish sort of color. This, we can see that it's a normal breast biopsy because you can see normal tissue surrounding the ducts. But this one, you can see that it's not actually that normal because you can't really even see the ducts or anything. So this is a case of cancer. Next is enzyme label. This is when an enzyme reacts with a chromogenic substrate. Chromogenic just means that it produces color. It's often used in immunocytochemistry because it allows the identification of a cell-bound antigen and its location by means of antigen-antibody reaction, and it's tagged by visible labels for a microscope. So basically what this is that if we want to find out where an antigen is in our body and to identify which one of what we are looking at is the antigen that we're interested in, we have the antigen. We need antibodies, so it requires the production of antibodies. And the antibody has a label attached to it. And what happens is that when the antibody and the antigen connect, when they react, the label will also react and it will become visible. And this can be seen through the microscope. This label is what makes it visible. What I mean when I say it requires the production of antibodies is, for example, what tends to happen is getting an animal and injecting it with the antigen that we're interested in, and it will produce the antibodies, and then we can collect those antibodies. Or we can even do that without the use of an animal, just collecting from the animal the cells that will produce the antibodies and induce antibody production. This is an example of an image used using peroxidase or NDAB. So this is human bladder cancer. Another type of labeling is fluorescent label. This is often as well used in immunocytochemistry, and it's also used in live cell imaging. This protein, called the green fluorescent protein, came from these jellyfish. As you can see, they glow around these areas particularly. And this protein is fused with the gene that we're interested in. So the gene that will produce the protein we're interested in is fused with this gene, with this protein, and is expressed in the cells or the animals that we're interested in. This allows for what we're interested in to glow. it also allows multi-labeling, so scientists have managed to create different colors, not only green fluorescent. In this case, we can see in cells that the mitochondria have been dyed glow in a reddish color. The actin glows in a greenish color, and actin is a major part of cytoskeleton, and the DNA glows in a bluish color, so it's the nuclei. The electron dense label usually happens in the electron microscopy, as the name suggests. In TEM, which is transmission electron microscope, it allows fixed tissue. It's accomplished by fixing tissue and embedding it in resin, and then cutting very thin sections and coating these sections with electron-dense stains. These stains are then bombarded with electrons, allowing these kinds of images, for example, mitochondria. These are very clear images with lots of detail. However, these images aren't 3D, but it's still pretty good. So we also have SEM, which is scanning electron microscopy, in which the surface of the specimen is coated with an electron-dense label. These other electrons are then bounced off of it, producing 3D images. In this case, we can see the moth antenna, and we can see it's pretty detailed. It is 3D, and these images are it's very good imaging. Sorry, something very important that I forgot to mention for fluorescent labeling is that, as it says here, it's using live cell imaging. So because it can be used in live microscopes, but it has become increasingly used in confocal microscopes because, as I've mentioned in the previous videos, confocal microscopes can allow us to see living organisms in 3D imaging. And because of that, we can see live animals without having to kill them in their actual functions. So it's very useful.